Okay.
And then if you want to go to school, you need to work on farm for certain period. So you need to wake up really early, finish your what you have been
Village
assigned.
life
And
starts early, definitely,
exactly.
with the sun. Yeah.
Yeah. And then when I moved to city for masters following PhD, we do a lot of partying, bar and everything. And then I was really ashamed that I couldn't sleep till late in morning, so I can't go to parties and have fun and drink a lot.
Okay.
Yeah.
So you are situated in Louvain-la-Neuve.
Louvain-la-Neuve. Louvain-la-Neuve.
Louvain-la-Neuve.
Yep. It's a
Okay.
French word.
Yes, it's mostly French at the southern part of Belgium, right?
Yes. Yeah.
Okay. So Louvain and Louvain-la-Neuve are different places, I think.
What one again? Yeah, Louvain. It's
Louvain
actually,
and yeah.
yeah, originally the university was same, and then they started having contrast over language. Dutch people, Dutch speaking
Dutch
community,
and French,
French,
yeah.
they decided to separate the universities and make it into two, French speaking and Dutch speaking. So the old university left at Leuven, speak Dutch, and then they started a new university around 74, I guess, 78, with the name.
The old one, I think it's in 1400s it was built.
Exactly.
Quite old, yeah.
Yeah, it's really old. It's really wonderful building too.
Yes, beautiful. I saw the pictures.
Yeah.
Good morning, guys.
Good morning.
Hi, Neha.
Hello, Dr. Kaushal.
Hi, Neha. How are you?
I'm good. How are you?
Awesome.
So, Soma, you're still preparing for the introduction.
Why?
Name.
Why? What are you saying? What happened? I don't know what you're talking about.
Okay, good.
Yes, there are some difficult names in there, French ones.
Yeah, I was seeing the profile and I was also quite confused.
Or you should use short names. For example, I'm sure you must be wondering about Fondelay research, something. FNRS.
In your Google Scholar profile also, I can see few.
French
I
words?
think it's not French words. There is some, I would say, strange kind of symbols coming up in between. I don't know.
That's
no, I couldn't see them.
Yeah,
it could be because the Google profile was
either it has translated it to something. It has encrypted or what, I don't know.
No. If you follow the Google page shared by Suvo, that usually comes up with the strange words. But if you Google directly and you go to the page, you will find
I went through Shuvo's link only.
Yeah.
So I was just checking, and there were some. I thought maybe the Google has translated the information and it has come abruptly like this in some encryption or something. So you are working on wheat now also, or you left the wheat rice science?
I moved. I wouldn't say move. I jumped a lot. So my master thesis was on maize. Then I moved to wheat. Then Arabidopsis, rice. Now I'm working with Arabidopsis and camelina, but sometime soon probably with rice again, because we have some results.
Yeah, I saw your profile. Like you have very, very varied concepts you have been working on.
Yeah.
Like microproteins, phytic acid. Then you have microproteins. Then now you're working on some TSOP and something
TSPO,
like
yes.
TSPO. That's good.
So how is your French coming along?
I haven't tried. I mean, I really want to learn at some point, because for teaching and stuff, you need to learn French.
Also for grocery shopping.
No. Google Translation works fine.
Oh, that's great.
Yeah, thanks to AI, it works fine. But my wife has started speaking, kind of introductory, and
Okay.
my daughter is in French school since 2 years, so she is really fluent now.
Oh,
She is
wow.
native.
Oh, she will teach you guys.
Yeah, that's
Oh.
what she does. As soon
But the
as we speak,
pronunciation and the writing for French is really different.
pronunciation is
Always I
particularly
pronounce
different.
wrong because
Yeah.
Shrikant knows French,
Mm.
and if he says me something, I can't spell it. That's
No,
for
that's
sure.
really, I mean, for us, it's really difficult.
If you don't know, then I can't ever spell in a single word if he says something. In
Yeah.
English, it's easy. We can still make those.
English is easiest language in world.
Yeah.
So I think we are starting.
Yes. Give me 1 minute.
Yeah.
You record.
YouTube.
My God, look at the comments. Good morning, good morning. Should I reply them?
No, we'll be handling the
Okay.
comment section.
No, just for sake of good morning. If they are wishing all it.
It's your choice.
Neha, question collection format is same. We again pasted the collected question in that shared docs. Select them and then we paste here over here, and use these brackets because otherwise they stacked and it's very difficult to where the question start and where the attendees name of the attendees. Okay, Shoma, ready?
Have you started recording?
I'm going to start. Are you ready?
Oh, you will say something?
Yes.
Can you please remember to?
Welcome everyone to Bioingene live webinar series on plant science. Today we have Dr. Kaushal. Shoma will introduce him. Neha will conduct the interview. Please keep your questions ready when during the question, before the question answer round, we're going to collect this from the YouTube chat box. If you have any other questions other than the today's webinar, please mail us. Don't ask here in that chat box. And I'm assure you during the question answer round, I will give the password and feedback link. Feedback link also available in our website. Then you go for the feedback and this link will self deactivated after 2 hours. So Soma, please welcome our guest.
Hello, everyone. Before we start today's webinar, I want to talk to you about a webinar on the 5th September that we are having. Hi, I hope you are all well and safe. I'm thrilled to draw your attention to an upcoming webinar on the Bioingene platform. As you know, that Bioingene tries to promote international plant science research But this is the first time that we are trying to address another important aspect. Those are soft skills. I'm sure you know a few of these soft skills and use them in your professional and personal life. Soft skills like communication, teamwork, collaboration, adaptability, openness to feedback, time management, stress management, work ethics, creativity, critical thinking, and problem solving are important for building a successful career. For example, I'm sure we have faced a common problem of time management. We, Bioengine, are arranging a webinar on our platform to discuss the importance of these soft skills. This time, we are also organizing a panel discussion. PhD scholars, postdocs, and scientists from UCLA, that is the University of California, Los Angeles, the University of Southern California, Los Angeles, the University of Calcutta, India, and the University of Florida, USA, will join the Bioengine team for a panel discussion. We will discuss the challenges we have faced due to the lack of soft skills and how we realized their importance in our careers. We are very fortunate to have Dr. Konkona Ghoshal, a former lecturer at the University of California, Los Angeles. Dr. Ghoshal has experience in teaching many innovative ways of soft skills in the USA. Her mission is to impart education in a holistic approach using research based learning and teaching methods. Her, vi- her vision is to prepare her students as lifelong learner to be successful in college and beyond. I personally request you all, the high school students, the college students, and the PhD scholars to join this webinar. You can visit our website or Facebook page to register. If you have already registered for this webinar, please inform your friends. Our webinars are free of charge and open to the whole world. Attendees will get a participation certificate upon feedback submission. Thank you. Now, coming to today's event. Today is our 14th webinar. I welcome you all to today's webinar organized by Bioengine. Bioengine is a non-profit organization created to promote plant research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are happy to arrange these webinars that are free for all. We want to continue doing so for all the students and scholars so they have easy access to knowledge on current plant science research. You can support this endeavor of Bioengine by donating on our website donation link. For today's webinar, we have received a huge response of around 3,000 registrations. We welcome all the viewers and thank them for their interest in Bioengine. It is our request to hold all questions for the question and answer round at the end of the presentation. You can type in your questions related to today's talk in the YouTube comment box. You can apply for a certificate of participation via the feedback link and password, which will be provided after the presentation. Submission of the feedback form is mandatory to receive a certificate. The topic for today's webinar is MicroProteins, novel tool to bioengineer future-ready plants by Dr. Kaushal K Bhati. Dr. Kaushal Bhati is a FNRS postdoctoral researcher at the Louvain Institute of Biomolecular Science, Catholic University of Louvain, Louvain-la-Neuve, Belgium. Prior to that, he was a postdoctoral researcher at the Copenhagen Plant Science Research, University of Copenhagen, Denmark from 2016 to 2018. He is the recipient of the Charge de Recherche Fellowship and Grant by Fonds de la Recherche Scientifique Belgium, that is the fellowship and grant awarded to the researchers by the National Fund for Scientific Research Belgium. He received the Special Research Fund Louvain 2019-2021 from the Université Catholique de Louvain, Belgium, Competitive Mobility Fellowship. He was selected as Plante Fellow and Network Leader, Plant Autophagy, American Society of Plant, like the American Society of Plant Biologist in 2019. He has many research publications in reputed international journals like the Genetic Society of America, Frontiers in Plant Science, Journal of Integrative Plant Biology, Plant Physiology, Journal of Experimental Botany, Scientific Reports, Plant Gene, Plant Science, Journal of Biological Control, etc. He has published many book chapters. He actively participates in research presentations and workshops. We are very happy to have him with us today. I now request Dr. Bhati to start his talk. Please, you can now share your screen. Thank you.
Kaushal, you start the screen sharing. Yes, it's coming.
So, can you see it?
Yes.
Okay. And this one, sorry. Let me reshare. Yeah, I hope it is clear now, right?
Yes.
Full screen?
Yeah,
Okay.
full screen.
So, good morning, everyone. It's almost 8 here in Belgium. And thank you very much, Shubho and team, for inviting me for this presentation. It's a wonderful initiative, I must say. And especially quoting the fact that you are providing information free of cost without registration to wide audience. It is just amazing. I mean, I really appreciate the concept. So I would start my presentation. I usually show this slide very early about my roots, how I developed myself from. So I came from a village close to New Delhi, although in UP, and where we involved in farming and stuff, especially wheat, rice, and other grains. And we love chapati, especially makka ki roti and saag. As you can see, my mother is cooking one. So close to my village, actually at backside of my village, we have a train track. So either way you walk, you reach the school. The other way you take train, you reach my college. And that's how we study a lot. We didn't have a lot of electricity at some point during my childhood, of course. Now we have everything. And then I moved to a not very known or famous college from bachelor, and then following that I moved Banaras Hindu University for my master's in plant biotechnology. And before finishing master's, I received a fellowship from Department of Biotechnology, Government of India for my PhD. And then I joined National Agri-Food Biotechnology Institute in Mohali. And after my PhD, I moved to Denmark, Copenhagen Plant Science Center, to join Stephen Wenkel's lab for my first postdoc. And currently I'm in Belgium for second postdoc on fund as mentioned by Soma, fund from FNRS. It's a fellowship and grant. And today I will talk about most sought project I was involved with Stephen. And he kindly allowed me to talk about this, which is really wonderful of him. So it's microprotein novel tools to bioengineer future-ready plants. And as you can see, you can follow the exciting result from Stephen's lab by following their Twitter handle from mine. I sometimes retweet a lot of good things. And then following Twitter handle of my current lab from Henry Botoko's lab. So I will keep it very simple because I can see the diverse background of listeners ranging from undergraduate to scientists. And I believe that a lot of things here might be very difficult to grasp for undergraduates, and they are really easy to grasp for scientists from molecular biology background and other things. But I will try to make it more easy and going to, so that everyone can learn something from this effort. So I will start with a very basic thing. It's called regulation of central dogma. So everything starts from a fragment of DNA gene, or you might call it loci if you are very classical. The DNA turns into RNA by transcription, RNA to protein by translation, and then the protein or group of protein, they control a certain phenotype or trait or a function or a physiological aspect of a plant or any organism in living world. And we know a lot about post-translational regulation. So post-translational regulation is when DNA is turning into RNA and RNA is maturing. At that point, you can do regulation of RNA quality or stability of RNA. It's called post-translational regulation. And then comes post-translational, transcriptional, and then translational regulation. So when protein is ready after translation, there are certain aspects how protein quality should get control. If an organism really want that protein or not, that decision also should come there after transcriptional processing. And there are tools, there are certain tools, those have been in use since start of molecular biology era to regulate aspect of DNA quality, mutation, recombination, and epigenetics. For RNA quality or control of RNA quantity too, there are tools like microRNA, small non-coding RNA, alternative splicing, RNA processing ways. However, for protein quality regulation, there are certain things, but there are not many tools those you can use to control post-translational things. How protein should express, do you want to control it? So everything comes before protein, which you can use to control if protein should form or not. But there are not many tools those actually target the post-translational regulation aspect of this dogma. And now there are new breeding tools, breeding technology, including CRISPR, ZFN, and other nucleases. They are now collectively called the new breeding techniques, and they cover DNA, RNA part, but again, not the protein part. So how we want to control the trait we want to introduce into a certain organism is all set of these things: DNA level control, RNA level control, and then protein level control. So everything which has been highlighted here by red color, mutation, recombination, miRNA, they all have been used as a tool to design certain aspect in living organism, ranging from bacteria to higher plants or animals or even humans. So as I have said that everything has been done very well at the level of DNA, RNA, but very few are there for protein, and that's why I want to talk to you about microproteins, which was work I did during my stay in Stephen Wenkel's lab. So let's start with what are microproteins. Now, I mean, the name says all, microproteins, so they should be small, of course. Microproteins, they have only one protein-protein interaction domain. They regulate and bind to a bigger protein with multiple domains. And then most of the known microprotein, they are actually regulators of transcription factors. It's not that they are only targeting transcription factor, but till now, whatever we have learned is the cases of transcription factors. As you can see in the figure below, this is a typical transcription factor structure. And if you see that it forms a dimer and binds to DNA. If there is a microprotein highlighted by orange color, the microprotein will bind to a protein-protein interaction domain of transcription factor and inhibits its binding to DNA. And that's how they were framed as a controller of transcription factor, or let's say negative controller of transcription factor. They sequester transcription factor into certain structures so that they don't bind their target DNA sequences. So how this breakthrough came, it's interesting way Stephen tells it. I mean, he coined the word microprotein after his paper from Stanford State. So he identified a feedback regulatory module by little zipper proteins. These little zipper proteins, they target SDGIP3 transcription factor genes. So, and I quote, encodes small leucine zipper containing little zipper proteins. They have domains. Those are found in Revoluta, Fabulosa, and Fabulosa protein. They are part of a big SDGIP3 protein family. And when you overexpress little zippers, you see that function of SDGIP3 transcription factor is reduced. So how this zipper protein works during wild type situation, small zipper protein binds to irreversibly to transcription factor. If transcription factor forms dimer, it's a normal situation. It activates the normal growth and flat leaf phenomena. When you have loss of function for zipper, you have too much of SDGIP. And when these SDGIP proteins form dimer, they cause upward leaf curl, as you can see here. But in other case, if there are a lot of zipper proteins, they voluntarily go ahead and bind to SDGIP so that SDGIP don't form dimer, and you see downward curl leaf phenotype. So that's how this phenomena of finding microproteins in plants start. It's not that they were never reported. They were reported as dominant negative before, or as a small ORF proteins, or just small proteins. But in this paper, review paper in EMBO by Stephen, he coined the term microprotein, and he introduced them very well. A group of protein that perturb formation of functional protein dimers. And again, I'm saying this part of science, microprotein, is quite evolving in his hand. And with time, you see the concept is evolving, the understanding is building. And here he mentioned that protein dimers are forming. And then if you have a microprotein there, these protein dimers will not form. Instead, one of the monomer of transcription factor will bind with the microprotein and get sequestered. It will not function anymore. And because the result of actions are analog to microRNA, which are negative regulator of mRNA, that's why they call it microprotein. Small proteins, those are regulators of protein and causing the post-translational control. And I must say, microproteins, as I mentioned in my first slide, are part of post-transcriptional control. Microproteins are part of post-translational control of protein. So how microprotein origin? There are two ways of defining microproteins. It's transmIPS and then cismIPS. So transmIP is a case when you have a small domain in mRNA or part of mRNA that cause single ORF to form a MIP. So there is a huge domain, large mRNA available already. And then there is independent ORF, part of that mRNA or part of that protein that independently translated and cause a translation into microprotein. It's called transmIP. However, complexity is even more for cismIPs. So cis is situation originated by 3 different events, or it could be more in future, you never know. So alternative splicing, alternative translation start site, and alternative polyadenylation. I will describe them in details here. So it's a full mRNA and then target. So part of this mRNA will undergo alternative splicing. The splicing will happen in a different way and then results into a smaller part of mRNA that codes for microprotein. So TSS here represent translation start site. What if case, if you have a translation start somewhere between mRNA structure, it will start from here and result into a microprotein. So, and then translation also turn turn at polyadenylation site, which is signal that protein structure should, protein translation should stop here. So polyadenylation, appearance of polyadenylation earlier than real polyadenylation site will result into a smaller ORF, and then finally a microprotein. And this all has been very well described in other reviews or maybe literature if you want to study or understand more how alternative splicing or translational start side, stop side works. However, in terms of microprotein, this is how till date we describe them. So as I said, the dimer formation by transcription factor and then inhibition of this dimer formation by microprotein, it causes into sequestration of complex or sequestration of transcription factor into non-functional form. The typical example here is zipper, which I already explained. So monomer bind to microprotein results into non-functional sequestration, but there are not only sequestration cases, there are more ways how things work. There are repressor formation where microprotein goes and bind to a protein where the target protein is part of bigger complex. And when it is part of bigger complex, it means that the microprotein will bind to target, and this complex, which was functioning well before, will not function anymore in a way that it was working. So it's called repressor formation. Cytoplasmic retention, if transcription factor is getting processed, coded in cytoplasm, and it should target into nucleus for function, microprotein can go and bind to transcription factor and stop it entering into the nucleus. And then ion channel, there are certain microproteins like VPU, those who binds to proteins on transmembrane region to their transmembrane domain, and then inhibit the ion channel functioning of these proteins. So how we find microproteins, we understand till now how they work, how they code, how we define them. So our colleague Daniel, he developed a bioinformatics pipeline. This pipeline suggests a follow-up how to identify microprotein from any known genome or set of proteome. How we filter the information is based on a certain characteristic, which we have defined before. Size of protein, microproteins are smaller than 140 amino acids. The sequence similarity to a bigger protein or a complex with multi-domain situation. The domain structure with target and microprotein should be very compatible in terms of sequence and amino acid composition. The protein domain that microprotein has should have a characteristic protein-protein interaction feature. And till now, there are 22 microproteins studied in Arabidopsis. Using this pipeline, which Daniel explained to MIPFinder, he filtered microproteins from different genomes, ranging from bacteria and then animals, yeast, as well as plant. And he used these 22 known microproteins as a quality control if he is able to pull down these proteins using the pipeline he developed. So following this pipeline, he identified that conservation of microprotein is not only limited to plant, but it is more wider spread. For example, you can see here Arabidopsis and mammals, Homo sapiens, Giamage plant, and then other life forms. And this is very complex. Yes, they are conserved based on protein-protein interaction domain. They are conserved throughout the life forms. And it is surprising good to learn in future if they also play conserved role. And that actually will be really, really interesting to learn for plant biologists as well as the life scientist in general. So if you look at the transcription factor family, the homeobox domains are most conserved across different life forms, following MYB transcription factor domains, and then BGIP in the end somewhere. So the overall message is these microproteins are pretty conserved. They function in a way that could be conserved across life forms and subjected to further research. So how this MIP finder helps? They identified a new microprotein complex, MIP1A, MIP1B, and then finally this study was published from Stephen Labs. So MIP1A, MIP1B forms a complex that regulate the flowering time in Arabidopsis by recruiting constant into a topless dimeric complex. So if you remember the introduction, I was saying dimeric, and suddenly it became trimeric complex. So that's how this area is evolving, and this is why it is very interesting to learn how a new concept evolves with time. So what are MIP1A, MIP1B transcription vectors? So constant regulator of a transcription factor, which is regulator of flowering time. It has these BBB and CCT domain. However, constant is not a single transcription factor. It has multiple members in there. And MIP1A, MIP1E represent a partial domain from this particular transcription factor family. And that's how the microprotein features looks like. So let's understand how it works. During short days inhibit GIFK1, and then that finally controls constant, which we were talking about. Constant forms a complex with BBX19. Whenever there is a MIP AB constant topless, it inhibits flowering time. And whenever you express or overexpress MIP1A, MIP1B transcription factor in Arabidopsis, it results into suppression of flowering time, or let's say it delays the flowering. And that's how the first MIP1A, MIP1B or multi-protein complex involvement of microprotein was discovered. So these microproteins A and B, they interact physically, as you can see from these two hybrid and the confocal images, and pull down. So dynamic situation, and the flowering is one of the most complexly regulated traits. So that's the question the lab asked, and all microproteins belongs to the protein classes. So this protein identification appears into an approach called synthetic microprotein. They are naturally occurring microproteins to regulate transcription factor, and they are well known, as I described in previous slide. But is there any way we can control diverse proteins using microprotein as a tool to control the protein functionality? And this paper in plant phase demonstrate how you can design microprotein, a synthetic microprotein, and target your protein of interest to use as a tool. So Ula, my colleague, it was her PhD project, and she identified that the microprotein percentage in Arabidopsis, it represents a lot hydrolases. Hydrolases are metabolic regulatory as well as other key regulator enzymes, the receptors and lyases. So she choose one candidate from each class, dicer-like protein, which belongs to hydrolases, bri1, which is receptor, and then cry1, which is a lyase. So I will here discuss how one of these cry1, one of these three, and I will focus on cry1 study, how they define, how the lab define how to design microprotein and how you should validate that. So cry1 is a protein with three different domains. She designed a MIP cry1 from pH domain. How this works, whenever there is a blue light, cry1 opens, and that cause deacetylation in seedling. If you overexpress a microprotein cry1 and expose it to the plant to blue light, there is a etylation. It means that MIP cry1 is causing non-dimerization of cry1 and resulted into better or less reception of blue light, causing etylation or elongation of seedling during early germination days. So again, validation part includes yeast to hybrid interaction. So she validated. And then As you can see here, this is col0 wild type, cry1 seeds, mutant seeds. So in blue light, all MIP1 overexpressors, they mimic phenotype of cry1 mutant. It shows that whenever you overexpress a cry1 MIP, it suppress functioning of cry1 protein at post-translational level and causing a phenotype which is similar to a mutant in wild type, dark as well as blue. So we discussed these approaches, how to use it as a tool, but we also wish to make it more available to public, and we wrote this paper about how to use microproteins and their approaches to build a tool in biotechnology. And we talk here about two different approaches, and this also evolved with time. Earlier we were only talking about approaches where there was a repressor complex was forming. That means microprotein was binding to a protein and then repressing its function. But we also propose here that a protein can also get activated by binding to microprotein, and that was one of the case that was reported in plant physiology paper. So we propose this pipeline, how to identify microproteins and then filter the information based on MIFinder information pipeline or software, then target identification for microproteins those you are interested in, or this could also go reverse way. You can identify first your target or a protein with multiple protein complexes which you find really interesting, and then you can use or design a microprotein to control that. Or there are ways you can use synthetic microprotein, or you can use MIFinder to identify if there is an interesting microprotein like sequence available in genome that could target my protein of interest. And then you validate your protein microprotein complex by biological function, for biological function using experiments that I defined before. And we will come to that part again in this end. So as a biotechnological tool, this paper from Stefan's lab defined first time how to use microprotein as a biotechnological control. So we design a microprotein from bbox2 HD MIP CDS from a transcription factor, which is HD1, a major master regulator of flowering in rice. And why we need to do that? So the biotechnological intervention, they always have a question and a need to answer. What was need here? In Nordic countries, especially Denmark, Finland, Norway, Sweden, the duration of days during summer is really long. It's like in Norway, there is always sun, as several of us know already. Even in Denmark, the duration of light is really long. We call them uh, strong long days. So strong long days as well as short days, they inhibit the flowering time in plants. And this sense of short long days or strong long days, it's sensed by machinery of transcription factors in plant, and that's how you signal plant to flower or not to flower. But this reception should be mediated by a better control that reflect how uh, plant should respond to lighting condition outside. And if there is a flowering, there is a yield. Better flowering, better yield, of course. So to cover up the situation of strong long days, what we design is an approach using the microprotein biotechnology to inhibit the HD1 transcription factor, HD1 transcription factor using HD MIP CDS, HD MIP HD1 microprotein. So you can see wild type during strong long days don't flower, while the mutant of HD1 flowers pretty well. And when you overexpress HD1 MIP as a control, there is not too many flowers or no flowering at all. But if you look carefully, that HD MIP1, HD1 microprotein 2 overexpressing lines as well as one overexpressing line, they started flowering just as like almost as similar to the HD1 mutant. So And if you look here, during short, long days, the flowering time, the heading time is flowering time. Flowering time has reduced a lot for HD1 mutant and the microprotein overexpressing and targeting HD1 in rice. So this behaves in a way that using a microprotein inhibition, you can control HD1, and during the extra long days, it cause a phenotype similar to short days and cause the flowering. So in a situation or a geographical situation like this, you can design your plant in a way so that you can control flowering time. But again, this is still limited to a particular trait the lab has been studying for a long flowering time. So we wish to move further deeper into the fact that if we are moving to crops, there are more challenges to address: stresses, pathogen attack, yield control, quality control. And we wish to expand this opportunity of using microprotein to control not just flowering, but also multiple diverse paths, metabolic engineering, for example. So we designed a project to identify microproteins in major crops, especially cereals. Cereals are being consumed across the globe very well. And we came up with this paper, which recently published in G3, Genetic Society of India, America. So what we did to refine our quality of microprotein identifying from crop genomes is we reshuffle certain things, we improve certain pipelines in MIPFinder. For example, the potential microprotein characteristic, we define that they should have a homologous interaction partner. They should contain single domain and high prediction instability index, which has been defined in our paper, how to use that. And then we made a filter of length less than or equal to 150 amino acid, should not have 10, more than 10 homologous in proteome, because plant genomes are really complex sometimes, thanks to polyploidy or the evolutionary recombination events. And then microprotein should exist on score less than 4 or equal to 4 on UniProt, because a lot of proteins we identified during genome annotation, they all drive information from UniProt database. So we also put a filter on target. There are a lot of filters. Those were coming with the previous microprotein finder pipeline developed in lab. And then we improvised everything. So using the newest MIPFinder 2 pipeline or software, which is now available on GitHub. If you go through the paper and you want to use the software, it is openly accessible. You can run the command line very easily on your computer. So we identify proteins, microproteins, small proteins, single copy, and all other information that we need to engineer the crop plants. And we targeted barley, rice, wheat, maize, and sorghum. These are very famous cereals. Those are being consumed and grown for commercial as well as household applications or consumables across the globe. So these crop we investigated, they are really rich in microproteins. We segregated information based on small proteins. Those are smaller than certain size. And then using new filters, we define if these small proteins are microproteins or not. And you can see all the information here. For example, rice, which is one of the most consumed grain. It is really rich in microproteins, and there are diverse pathways these microproteins are covering. And certainly some microproteins are single copy. They make more confident cases if you want to study them using CRISPR or maybe other ways. And then the number of homologs they have. So let's go back to this proposal we put forward, how to study microprotein. So we invite everyone, you should go through paper and then follow up that has been done to use microproteins as a tool to study or design new crops or metabolic engineering. So again, there are certain things to understand. Alternative transcription that also not limited to splicing, but also polyadenylation or early stop codon, small ORF, how to identify these things. Then we have two different versions of MIFInder. The modified one, which recently published in G3, was MIFInder 2, openly accessible. Everyone should go and try that on Genome of Interest, not only on plant, but also on yeast or any living organism which has been sequenced, or you have the proteome information for that. Overexpress that and validate these things by protein-protein interaction assay, loss of function development by CRISPR or knockout or RNA, whatever you prefer. And then experimental analysis to validate if things that you designed are really working. And they are not, the information is not limited on to this. There are a number of other studies coming up soon from Stephen's lab, which I can't talk about here, unpublished, of course, but they will be out soon, and they will show how to use the microproteins to control or design metabolic engineering, or maybe quality trait development in crops, or regulation of certain things. So here I would like to stop and thank everyone. I would like to acknowledge Stephen for his consistent support during my stay, as well as even after that. He's always a good mentor. He's always mentoring, suggesting ways to improve science and general perception of science, in my opinion. And Valdoko, his new colleague in Stephen's lab. And I would like to acknowledge the past colleagues I had wonderful time with them: Enko, Bin, Daniel, Esther, myself, of course, sorry, Shinyong, Tanai, and Wendy. And now I would like to invite you to ask questions if you have some.
Yes, we have many questions, but I understand that this field is very new. So most of the questions is most of the either unpublished data or belongs to the new discovery. But there are some questions I collected from the YouTube.
Sure.
I just wanted to paste here. You go through it there. If it is possible to address, then please address them.
Should I go through them?
Yes, you can choose, or you can go through them all. We have more. I just pasted few, and we just keep ready for us.
Okay, let me try. Okay, I'm going through the double screen problems. So where should I look for questions? Have you pasted some? Hello.
In the chat box, have you got the questions? In just beside the blue button, screen share, and then there is a polling, and there is left and right is a chat box.
Do you mean on Zoom?
Yes, on Zoom.
Okay, yeah, sure. Chat. So the question is from Bablu Gupta, which says, can all microproteins work? No, I mean, if your way of identifying microprotein is really sure, they should all work. Although I would always suggest that the biology is science of uncertainties. So you should always test everything experimentally before predicting anything or going through. So for example, we screen a lot of things and then we funnel down into limited stuff, which is really confident one. They all should work fine, providing that you validated everything beforehand. I hope that answers your question. So next question is by Asmita. can microprotein play a role in detecting viral disease? Of course, this is really new area which hasn't been explored very well. And I would suggest everyone should go through the papers Stefan is publishing, or I have shown in my slides. There are certainly roles associated with the pathogenicity for microproteins. Only problem is there are not too many labs in world those are working with microproteins, and the resource is there, information is there, not too many people to play with. Not everyone can go and work with the, if we are working with flowering time, you can't go and work with the viral diseases. So sure, you should go and read more about it. Next is with the Juma Datta. Can transcription factors be considered as microproteins? No, transcription factors, if they have been through the certain things, for example, alternative splicing, and they lost a domain and retain a protein-protein interaction domain. So how things work is transcription factor have two domains generally, typically, protein-protein interaction domain and a DNA binding domain. Imagine if situation is there where during alternative splicing or polymorylation site or by processing a protease, the DNA binding domain got depleted or deleted, and the domain left is protein-protein interaction domain. It is the case that I showed with the transcription factor studies I showed before, for example, MIP1A or ZIPR3. The protein interaction domain was there, but DNA interaction domain was lost. So that is the case where transcription factor are being targeted by microproteins. Yeah. So what are the tools to identify microproteins in animal cells? Pipeline designed by Daniel is applicable. Yes, it is applicable. Even the new pipeline I have showed recently in G3 paper, these two pipelines works very fine with any genome, you know. There are, even in Daniel's paper, there are a number of microproteins reported already from mice as well as human. And in fact, he also mentioned something related to cancer. This is another huge part. So number of labs, they are identifying small regulatory proteins by naming, calling them small proteins or small ORFs. And there are a lot of cases, such cases in cancer, cancer situation where proteins or ORFs are undergoing alternative splicing or other things, and then forming a smaller version of protein, and they could be potential microproteins. So Sanjana, how to identify microprotein? I already mentioned a number of papers, but if you still want to go through, I would suggest if there is an organism that we have already studied, I would say just go through the paper in supplementary file. For example, in recently published G3, there is a supplementary file, identify microprotein from these 6 genomes that we studied in plants. So you can directly Google, not Google, I mean directly search for your protein of interest there in the sheet we published. Are there identified microprotein? I mean, they are not identified and validated experimentally, but the recent paper in G3, it suggests a huge implication of microprotein during drought and salinity stress. So I would suggest go through paper and you will find a lot of interesting things, especially in plants. Yes, you can, Dr. Satyapal Singh, yeah, you can diagnose. So what I propose sometime, I mean, I also have good belief about that. During disease situation, certainly there are microproteins are being formed. If one could identify them, you can use them as a marker to diagnose the disease. The only factor is, again, experiments, validate experimentally, and then once you have a good candidate, you can always use that as a marker to diagnose the disease. Kostova, do microproteins involved in silencing of genes? If yes, what is mechanism? So I already told the difference between post-transcriptional and translational. So silencing of gene is actually silencing of mRNA that happens post-transcriptional using small RNA, miRNA, or artificial miRNA. Microproteins target proteins. So post-translationally, not silencing of gene, sorry. Neha Tiwari, are microproteins present in money plant? Why? they increase very rapidly. Awesome question. I, I, I wouldn't be surprised. They grow because they have physiological way of growing this way. It's just very naive to say anything about it. But if you know money plant has been sequenced, why not you go through the genome and apply MIPFinder pipeline to identify if there are microproteins associated with this quick growth of money plant. The only scientific way to do that or validate that. So more questions. Dr. Satyapal, again, can we diagnose the disease of plant? Yes, as I mentioned before, identifying microproteins expressing specifically during disease condition and then validating experimentally, that validated microprotein could easily be used as a marker. I mean, you can develop antibodies against it, do ELISA very easily, and through the crushed sample that you prepare from disease plant. I mean, it's a way to, what I think it's a way to develop something important or interesting in future. Harishankar, is microprotein applicable in breeding program such as paddy? Yes, of course. That's why we published this G3 paper, because we want breeders to get involved. And so I'm sure Stephen has a lot of things going on with the breeding companies in future. I mean, in talk, but certainly things will appear in future. So if you are working with paddy, I would suggest you should look at the microprotein list we published in recent paper. And if you have certain candidates, please identify your candidates and I would be happy to help you taking them further. So Kostov, do microproteins involve in silencing? Okay, no, I already answered that. Can we develop a microprotein modified plant that may be used to reduce contaminants? So I showed that there is a case where microprotein control ion channel. If you know certain ion channel, those who took up soil and took up contaminants from soil through root, why not? You can design a microprotein, either naturally identify that or synthesize one using the approach mentioned in plant-phase paper, and then target your ion channel or a transporter, for example, using microprotein, overexpress that, and then test your plant over the contaminant soil. You could see something. I mean, again, you can talk a lot of hypotheses, a lot of ideas over plant biology and experimental biology, but again, everything funnels down to how concrete experiments you design to validate something. So Ibrahim, microprotein often set to as a decoy, thus interaction with the target result in reduced activity. What is the truth? Yes, this is a quality question, Ibrahim. Thank you. So decoy concept is pretty much similar to microprotein. They reduce activity, and it is actually true. I would suggest you to visit Joshua lab in Yale. He was colleague of Stephen, and they both did postdoc together. And Joshua moved into decoy part, and Stephen started microprotein. So there is an overlap between these two concepts, protein decoy and microprotein regulation, and they both are interesting, I would say. So what are application of microprotein in synthesis of secondary metabolism? Shani has this question. So the application of microprotein is in metabolic engineering, and evidence will come soon in future from Stephen's lab. And I am sure if you go through the list of microprotein we published, you will find number of microproteins targeting metabolic engineering, metabolic pathway proteins, especially the key enzymes and the step regulators. And those proteins could be explored very well by community. For example, if you want to take one of most confident protein from our list, you are welcome. And please come back. I would happy to help. Or you can always write to Stephen. He's really good replying. at replying and helping people. Falguni, microproteins can combat metal stress. Yeah, I would second my answer that I answered to Rajni about reducing contaminants. Metal stress can be combated very easily, but again, it's a pipeline that I showed on last slide and somewhere in between, how to identify and validate microproteins. There are a lot of microproteins unexplored, and this is what inspired me to do this presentation. Instead, I left the lab, but I always feel close to this topic. And yeah, there are opportunities there in regulating metal stress in plant. So Shanmugam, what is average size of MIP? Average size, I would say the pipeline we propose is 140 amino acid to 150 amino acid, not longer than that. No, base pair, actually, why I'm saying amino acid. Okay. I mean, yeah, I propose, I shows that in a slide. Microproteins can combat, yeah, she replied, I replied her. What are consequences of overexpressing? What are consequences of overexpression and loss of action of microprotein? So consequences are, for example, they all depends on which microprotein we are talking about. In case of zippers, which I showed the first case earlier, it says if you overexpress and then you knock out, there are different ways of leaf phenotype. For MIP transcription factors, if you knock out both MIP1A, MIP1B, there is no phenotype as published in paper. And if you overexpress MIP1A, there is a delay in phenotype. Can we identify microprotein in M? Oh, virus genomes are really small, and I can't comment on it. I mean, what is happening to virus genome after entering to host? It's really uncertain. It's really complicated to answer this question, but I would suggest if you go through paper, you might find a number of things to address, especially with the viruses and pathogens.
Thank you, Kaushal. There are no relevant questions.
Okay.
I request Neha, Dr. Neha, to conduct the interview to Dr. Kaushal.
Thank you, Shiva. Thanks, Dr. Kaushal, for such a great talk about a novel concept coming along in plant biology. I'm sure it will have good implications in future plant science research. So now we are moving ahead to the interview section of today's webinar. Today is our 14th webinar of the series, and we are honored to have with us a very distinguished scientist, Dr. Kaushal Bhati.
No, I'm not distinguished.
We would like to know more about you, and I would like you to describe your journey so far as a scientist.
Okay, so I mean, I haven't traveled a journey yet because I'm pretty young to say these things even now. But what I have learned is through my teachers, mentors, and friends a lot. And I mean, these are learnings I always appreciate, and I admire them, their time they give to me during teaching or discussions, during tea time, for example. That's another biggest aspect of being in research. Yeah, so I mean, what I followed is typical paths everyone follow. Is I mean, I wanted to get involved in research, of course, but before that, I wanted to join Indian Army.
Okay.
At some situation that didn't happen because of a few things. So, and then my father wants me to follow medical, which I find boring. So there was a clear path around my high school that I want to go into research. And during bachelor's, I was more interested in human physiology and things, more into kind of medical research or biomedical sciences. But things didn't happen that way too. So I followed, I gave number of entrance exams and I got selected into the BHU plant biotechnology program. It wasn't my first choice, but I had no choice either, so I joined that. And during masters, I got fellowship. So I wanted to join a certain group I was really sure about. So I joined them, and things didn't went really well. So I switched to other lab where I finished my PhD with the Dr. Ajay Kumar Pandey. And I'm really in gratitude for what I learned in his lab, including personal life, professional life, and then scientific attitude, of course. And following that, I moved to Stephen's lab, and he was kind enough to offer me postdoc even before I finished my PhD. So he booked my ticket in advance. You finish your PhD and fly to us. So, and everyone, my faculty in Nabi, where I did my PhD, in Punjab University, they helped me a lot to get through fast the PhD process and as well as defense, so that I can catch my flight right next day of my PhD defense. So
that's great.
so I flew to Copenhagen, and then I joined the Stephen's lab, which was really great stay in terms of learning, meeting a lot of good friends, and the scientific aptitude, of course. I mean, that's what I'm saying. For my journey, I always dedicate my journey till now, which is not journey yet. I don't know how far I will go in future. But this all goes to people I have discussion with, the people who give their time to me as a teacher, mentor, friend, family, of course. My parents, brothers, sisters, wife, and now daughter. Yeah. So post microprotein lab, I joined Henry Batoko lab here in Louvain. And here we are doing now really cool and exciting stuff related to plant autophagy, as well as some other side projects. Those are also should get published soon. And again, I mean, I have really good journey in terms of having good mentors. I'm really satisfied here too, again. So thanks to Henry. And we also got cool grants to work and to support our research here in Belgium. Yeah. So, I mean, there is no pathway that I followed. It's just that I believe in flow. Whatever I keep getting, I keep following. I didn't wish too much. So it's everything which I have is what I get.
Okay. Yeah. Thanks. My next question is, how can we translate our knowledge of microproteins into food security?
Yeah, that's actually really cool question. So food security has, in my view, two aspects. First is enough yield to feed everyone and enough nutrition to make everyone healthy. So during my PhD time, I worked with nutrition quality, including reducing anti-nutrient phytic acid content in wheat. That was also part of nutrition security, I would say. But in terms of both aspect, food security and nutrition security, microproteins could be really potential. The paper we published recently, it talks about series of microproteins targeting different pathways, which I answered also during questions. For example, there could be microproteins, those are controlling yield or knocking them out using CRISPR, or overexpressing them could change the things we are seeing now. For example, the yield loss due to drought. Nowadays, there is a lot of drought in Europe, as well as not enough rain due to global warming. The temperature is rising. So the food security is a bigger aspect of seeing bigger problems, or how bigger problems are influencing our food, not by quality, but also by quantity. So the biotechnology in general is also answering these questions, but MIP as a tool could be subset of that bigger efforts that biotechnologists are doing to secure food and quality for us for future generations.
Okay.
Yeah.
So coming to my next question, which sectors of plant science research and agriculture will prosper in the near future? according to you?
Food security.
Like
I
some,
mean, you know,
do you specify some technological innovations or some?
I see. I mean, there is no such God technology except CRISPR till date. Before this, RNA was something that you reduce or regulate negatively certain mRNA and generate quality trait. For example, happened in development of different commercialized varieties in soya bean, for example, or other crops. But yeah, CRISPR could be the breakthrough. I mean, it is already a breakthrough. I wouldn't say could be. We are already seeing a lot of magical things CRISPR is doing.
Mhm.
But again, agriculture is, or food and agriculture aspect is very crucial for population on Earth. And these technologies should help securing good food and nutrition quality.
Mhm. Okay, coming towards the end of our interview, I would like you to give few words of advice to the young research fellows out there.
Oh, I'm young.
You're
so,
young too, but there are
so
more
I,
younger generations
yeah,
seeing you,
uh,
listening to you, and taking
okay.
up inspiration from you. So we would like you to
So
give
I,
advice
I
to
mean,
them.
I don't want to sound old by advising this, but whatever I have learned till date is, if you are at undergraduate level, read a lot of books and learn the basics, because they goes along till you die, especially good books. And spend time in library and enjoy time with friends. So and then I mean, in India particularly, there is a pathway that everyone follows. For example, do a bachelor, join a big university for masters, join a research institute for PhD with a big name mentor, get fellowships at every stage. That helps your CV. And then after publishing a huge amount of paper, go behind all these awards and stuff. They looks good on your wall, which I don't have. So and
You'll soon have.
and then for masters, I would say they should focus more on developing the question ability. Asking question is a really big thing, not answering question. So what we focus more on getting superficial knowledge, and then we want to quickly move on to something which I really find offended. I mean, for my personal sake, I digged a lot of books. I mean, all of my friends, actually, all of my circle, we read a lot of books, every book possible in molecular biology. And that really helps. There are faster ways to do something, but there are more interesting ways to do something. If you want to retain in research, you need to learn how to question and how to find answer. So at masters, everyone should learn how to ask question. During PhD, you should learn how to answer question. And of course, talking to people. That's another biggest thing I would say should learn at PhD situation. Talking to people, making network, and then learning how to deal with the stress, of course. And yeah, network, dealing stress, learning how to answer question. That's enough to learn as a scientific thing. For postdoc situation, I would say we should know how to lead. We should learn how to lead things, how to take initiative over things. And then because everything which we have learned is now time to apply that. At some point we will be leader, group leader or something. We know how to talk to people, which we learned. We know concepts which we learned during bachelor. We know how to answer questions. We know how to lead. So the step by step you learn, you become something. And on philosophical part, I would suggest everyone should be feel grateful to the people who are helping you, people who are around you. Everyone should help each other, because this career path is really stressful. It's really struggling. It goes really long away. You really need companions to go along with you. And when you don't have companion, you start losing the personal peace, inner peace. Yeah. And yeah, whatever you do, unless you have a deadline coming, always choose life.
Sure. With such a nice, inspiring words, I will hand over it to Shivu. Thank you, Dr. Kaushal, for your kind words and your time. Over to you, Shivu.
Thank you, Neha. Thank you, Kaushal. As you say that is the reason in my mind that we should learn some soft skill also, because we realize during our PhD lives and postdocs that it is also a part of our life. That's why I'm interested to introduce this. And there are some questions that which books
Which
in
books?
in molecular.
Okay. I mean, I personally like Voigt and Voigt for biochemistry, Cooper, and then I mean, there are a number of good books which I can't recall anymore. For example, Leininger, everyone read that. Everyone has personal copy.
I have a question. Do you read Albert's?
Ah, yes, I did that.
Okay.
Yes. I mean, we couldn't afford everything. So what we used to do is instead of buying original copy, we were buying photocopy of them.
Xerox.
And at my home, at my parents' home, actually, I have a almirah full of photocopy books, which I still go and read sometimes during holiday. It's really fun.
And I'm suggesting these fellows, those are asking for the books. There are lots of books. If you need someone, it's always available in the library, and ask your librarian. I think he or she missed a lot. You guys, you go and ask the librarian that which books we
actually having relationship with librarian is wonderful thing. I always had so good. They offer you tea whenever you are alone. They offer you free books to read.
And very much foreign author books come with the CD and DVD player.
Exactly.
Okay. Most of time they are kept in the library, not in the bookshelf. So you can ask their CD and DVD. So you can portable and read in mobile, iPad, or anywhere. And sometimes books gives the keyword to register in the website. You can register and learn.
Another
asks us to, we have a full of drive with the good books. If you need then ask us, mail us, we can share with you. That a lot of books are available, and it is also help you guys.
For molecular biologist, I would suggest everyone should read Genes series. I mean, Gene book is a series. It's so wonderful. I mean, there is nothing like Gene.
Yes, and I would add that Gene 8 may be very helpful to beginners. If you start Gene 11, that it may tough for you. So start with Gene 8. It's very, very handy and very helpful.
Yes.
So I'm again thank to Kaushal that you come and support. It's one type of support also, because
of course.
It's a platform. Many comments are there to appreciate your talks, also appreciate Bioingene. But I thank all of you, including you, that you come and give your talk. We are very happy in future if you do your if you wanted to share more of your research from your lab.
If I publish more soon.
Yes, obviously. And best of luck. You can publish more, because this field is new, and I think lots of coming in that field. So I always wish you that you could publish more and more.
Thank you very much. I wish really good luck to Bioingene team, and you are doing wonderful. Thank you very much. Have a good day ahead. Bye-bye, everyone. Bye.
Bye-bye. Thank you, Neha, for supporting, collecting the questions, correcting this all, and helping to successfully conduct this webinar. Thank you. I just wanted to say thanks, and thank you, Neha. Thanks, other member of Team Bioengineer to participate today's webinar. I wanted to give a small reminder for our upcoming webinar So today's password is 85584. I'm sure everyone got this information. Password link for the feedback also given here in YouTube chat box and also available in our website. Our next webinar, these are our next upcoming webinars. Registration are ongoing from our website. Please go and register. Today's password is 85584. I'm going to end this meeting, end this webinar, stop the broadcasting. I pasted already. I already given the feedback link and password in YouTube chat box.